All right, folks, so today we are going to talk about vision. I know most of you are thinking this was a class about physiology of behavior, and you're wondering how vision falls into behavior, right? I mean, if you're just looking at something that seems rather passive, right, Tyler? Open my eyes and things happen, that's great. But we're going to spend this week and next week talking about the sensory systems. So we're going to talk about vision today, and next week we're going to talk about everything else all in one. And for those of you that think, well, that's really giving those other sensory systems kind of the short end of the stick, well, that's probably true. There are some reasons why we're going to spend so long on vision. Vision is for humans at least, it's sort of our primary sensory modality, right? I mean, most everything we do is pretty heavily based on vision. Not too many folks are smelling their way around the world, right? So some people might try that, but it's not going to be very, it's not going to be that great, right? The other thing is we know a ton of stuff about vision, more than we maybe know about some of the other sensory systems. On top of that, basically the same basic principles that apply to vision are going to apply to the other sensory systems as well, right? So once we get the handle on what's going on with the visual system, then we can apply that to the other sensory systems, and we're really just kind of changing things a little bit, right? So we'll talk about the eye, we'll talk about brain regions involved in visual processing, a little bit about color, spatial vision, orientation, and movement. I'll tell you what's important. Don't get too bogged down with some of the details, right? Anybody in my sensation and perception class? No, nobody's in that class? Great. I didn't want any of you in there anyway. That's why I registered it that way, Abby, just to make sure. If this really excites you, you should think about taking that class, because we go into a lot of detail on different sensory systems. All right, so the first thing we want to do is set up a couple definitions. We want to talk about sensation and we want to talk about perception. These are two different processes, okay? Sensation is really just the cells of your nervous system detecting a stimulus from somewhere in the environment, right? So in this case, we're thinking about your eyes, we're thinking about your retina. You've got photoreceptors that are collecting bits of light, and they're going to respond to that in some way, right? So they're going to take that information, that light information, they're going to convert that into an electrical signal. As we learned, a week or two ago, your nervous system runs exclusively on those electrical signals. So anything that's in the environment around us, we have to convert that. We call that process transduction. Sensory transduction. We're going to take some stimulus other than electrical input and turn that into the electrical signal. Let the brain understand. It's a very simple process here, the sensation. It's just that stimulus interacting with the receptor, interacting with that specialized cell. Okay? Now, how you make a conscious experience out of that, how you interpret that, that all falls under perception, right? Today we're going to talk about wavelengths of light. There really is no such thing as red light or blue light or green light. There's light with different wavelengths, and we perceive that as red or blue or green, right? And so that perceptual process is separate from the sensory process. Often they're difficult to pull apart, but just be aware that there are two things. All right, if we're talking about vision, we're going to use words like hue sometimes. That's the dominant wavelength of the light. We might talk about brightness or intensity, and then saturation, which is going to be the purity. Anytime we're talking about a sensory system, we'll talk about sensory receptors. And these are going to be specialized neurons that are only going to be tuned to detect a very slim category of physical events, right? So when we're talking about photoreceptors, we're thinking about things that are only sensitive to light. Audubon actually goes a step beyond this, right? So there are photoreceptors only sensitive to certain wavelengths of light. We'll talk about what that means in a few moments. All right, we already discussed sensory transduction. The only other concept that we really want to sort of have under our belts right now is this idea of a receptor potential. Now, previously, Paris, we talked about action potentials. We talked about membrane potential, right? You guys have been telling all your friends about this because you were so excited about it, you just couldn't contain it, right? And I know It's been blowing up on social media. I've been following it. So, the receptor potential is a little bit different, right? These are what we call graded potentials. If you remember, that action potential was an all or none event, right? We were cruising along and then all of a sudden, wham, we've got us an action potential. That's the end of the story, right? There was no way once we started that action potential to stop, okay? It was an all or none event. Receptor potentials are different though, okay? These are actually graded, right? So these things will go up and down, up and down, up and down, right? As things get excited, they'll move up a little. As they get inhibited, they'll move down a little. That's what we mean by graded. This class is a graded class, right? You can score somewhere between 0 and 100%, right? If this was a pass-fail course, anybody ever taken a pass-fail course, credit, no credit, right? That's what we would think about for an action potential, right? That's an all or none. You'd either got all the credit for that class, or you got none. Right? In this class, you can get some of the credit. You get 75% of the credit, right? That's a possibility. How many of you are hoping for 75% of the credit? Anybody already setting their expectations there? No, nobody's going to commit to that. Always like to ask. All right, this is the electromagnetic spectrum. Okay, this is not to scale. Right? Not to scale at all. Okay. So you have this thin sliver of the electromagnetic spectrum to which your eyes are sensitive, right? That's somewhere between 400 and 700 nanometers in wavelength. So when we're thinking about a wave, waves look like this, right? We measure from peak to peak. That's our wavelength, right? Okay. That's a lambda, or my attempt at a lambda, by the way, for wavelength. So we're measuring peak to peak. That's wavelength. The wavelength here is, 400 nanometers. For your purposes, you just need to know that's super, super small, right? Okay, it's not the smallest, usually, but it could be smaller, right? How many of you are familiar with television and radio? That comes in waves, right? Those waves are huge. Those waves are like the size of a building, right? Or massive, okay? Low energy, hopefully, right? That's what we've been telling you for years anyway. Low energy. Bethany, you got that. That was funny, right? If not, you were all, who knows, right? So it's low energy, right? They're huge waves, they're low energy. Nobody's walking out and getting knocked over by a blast of a radio wave, right? It just doesn't happen. As you move to smaller wavelengths, you can start to pick up more energy, right? So we have infrared waves. How many of you have heard of those? Those are awesome, right? Anybody have a remote control device or some of those run on infrared, right? the older devices would. Then you move into the visible. We call it visible. That's because we're very egocentric, right? It's only visible to us. It's called visible light. You keep moving on down the scale, you get ultraviolet rays. How many of you are familiar with those? This is a great discussion to have around that third week of March, right? The ultraviolet rays, that's spring break. So it's a good time to think about those things. X-rays, familiar with X-rays, right? If you ever need to see bones or other things. And then gamma rays, those are always fun. Not really something you guys need to worry about. Now, we call it the visible light spectrum, this kind of wedge in the middle that we've expanded. You can see, there's a problem with the screen. This is sort of the bluish purple end, and we're headed over here to red at those longer wavelengths, right? There are some species out there that are actually sensitive to things outside of our visible light spectrum, which is kind of cool, right? There are certain birds and insects that are sensitive to ultraviolet light. That's pretty cool. You can take images of some flowers using ultraviolet light. You'll see that they reveal these sort of interesting patterns that aren't visible to us under regular wavelengths of light. They're only visible to those species that have receptors for that. And how many of you love pit vipers? Anybody's a pit viper fan? Okay, that's too bad, because we're bringing pit vipers in next week. So just get ready for that. Why do they call them pit vipers? You dig a pit, you put the vipers in, you push people in there you don't like. That's why they're called pit vipers. No, they're called pit vipers because you have these little pits kind of back on their head, right, that are actually sensitive to infrared light, which is cool. There's a guy in Germany who studies these sort of odd sensory systems, right? He's studies these frogs that are sensitive to like waves in the water, but he studies these pit vipers. And he does this interesting study. You can look at it, it's on the internet. First you have to grab the snake, right? You always grab the snake by the neck. I don't know if you knew that. You think like, where's the neck of the snake, right? It's like the whole thing. Make sure you want to get as far up on the neck as possible. Don't grab way down on the neck. You're going to get bitten. So you grab the snake, and then he does this brilliant thing. And he uses this really expensive and complicated scientific device, Anisha called electrical tape. And he takes electrical tape and puts it over the eyes of the snake, right? So now the snake can't see, right? Which is really great, because if you ever really want a snake mad, you put electrical tape over its eyes. That's definitely what works. So then, likely he's got to put the snake back in a cage, right? Which I think the maneuver for that is this. It's kind of like a shot put thing, but aimed downward, because you don't want to like just gently put the snake down, because again, it can't see, you've strangled it for a while. What he does then is something really amazing. He lets a rat loose in this cage with this pit viper who is now essentially blind, right? Because he's got the electrical tape. The cool thing about this is the pit vipers can actually just right on top of those rats and just attack them because they have those cool infrared sensors, right? And so they can kind of map out the infrared signal coming off of that rat, even though they can't see. I don't know how he does the next part of this, Mary, which is removing the electrical tape. Seems to me that would be far more difficult than putting the tape on the snake, but somehow the tape comes off as well. Okay? The rat doesn't make it, but the snake's fine. The snake handler, I don't know. Right? I'm assuming there's some puncture holes from time to time. So, other species are sometimes sensitive to other parts of this electromagnetic spectrum. We're only going to be really concerned with that visible spectrum from around 400 to 700 nanometers. All right, so you've got a retina that's on the back of your eye. How many of you have been to the eye doctor? Yeah, I can take a wild guess as to how many of you have been there. When they shine that bright light in your eye, that's not just to torture you. I mean, it partly is, but it's also partly to see the back of your eye, the retina, right, because they want to look for retinal health. This is particularly important for certain disorders. One of those disorders being aging, right? That's the disorder you're never going to outrun. No cure for that one, so just hang in there. Diabetes will also cause some degeneration of your retinal health, so if you're diabetic, definitely need to get your eyes checked more often than if you're not. But there are some other disorders that have macular degeneration, other things. You can think about those some other day. Embedded in that retina, you've got these guys called photoreceptors. These are receptors that are sensitive to photons of light, right? So anytime light comes in your eye, it comes in through that awesome thing called the pupil. It's going to hit on the back of your eye. It's going to activate those photoreceptors. You have two kinds of photoreceptors. You have rods. They're so named because they're shaped like rods. Very creative. And then cones, because guess what? They're shaped like cones. They actually do two different things, though, when it comes to light. Rods are pretty sensitive for low-intensity light. Cones really need a lot of light to get them activated. And cones are for color vision as well. How many of you have ever thought you saw something at night and then when you looked at it, it disappeared? Yeah, it happens all the time, right? Thanks, Bethany, for being brave, raising your hand there, right? And then you get a little nervous and you start looking around like, what is that? And then it disappears. And of course you make up a great story like it must be, you know, like the ghost of your great-grandmother or something coming back to haunt you because you messed up her biscuit recipe. It's a true story. So what's really happening is whenever you look at something and you really want to examine it in detail, you should always put it like in the center of your vision, right? So that it falls on something called the fovea, which is this spot in the middle of your retina, sort of the middle, which is really tightly packed with a bunch of cones, right? So you've got super good vision there, the best you're going to have anywhere else, and it's really sensitive to light, colors. Humans being diurnal, which means we're up during the day instead of nocturnal, right? Which means we're kind of out and about during the night. So we're really going to use those cones, right, to kind of get our way around. Now the problem is when you see something out of the corner of your eyes at nighttime, your cones are pretty well useless, right? But what's not useless are going to be your rods. The thing is, all the rods are out in the periphery of your retina. None of those are in the middle, right? So when you catch something out of the corner of your eye with your rods, and you're like, oh, Okay, but I'm going to see what that is. And I move my eyes over there. I then put that, whatever it is, onto my patch of cones there in the fovea, and it disappears because my cones are no good at night, right? And then you look away and guess what happens? It reappears, right? Because again, it moves to where your rods can pick up that light, pick up whatever that object is. So you're constantly looking back and forth. You can do this if you want to test it tonight, go home, turn out the lights in your house. Wait until your folks who live there with you show up and then jump out and scare them. Once that's over, you can all sit down and give this a try, right? You just kind of like look around, see these things up here. This is a particularly important thing to know if you, how many of you go outside at night and try to look at things in the sky, like stars and so forth, right? Yeah, it's a particularly important kind of thing to keep in mind, right? But look at that, there's an eye. You've got muscles that move your eyes. That's exciting. And that's about all we're going to think about that. We might talk about a couple other things, but that's it. So here's your eye, it's cut in half. That sounds like that's going to be really pleasant. Anybody ever dissected an eye? All right, great. Yeah, was that a cow eye? Usually it's a sheep eye, really? Usually they do cow eyes, they're a little bigger, but a sheep eye will do the same thing, right? So you cut that thing open, full of squishy stuff. There was probably, looks something right here that looked like a butterscotch candy, right? Yeah, that's the lens, that's pretty cool. If you cut through that carefully, it kind of flakes off. I don't know if you did that or not. You have to watch out though, because it'll shoot right out and get you. That's the lens. You move that guy up and down, right? Some people are better at that than others, and as you get older, you're not going to be as good at that, right? So some of you might have noticed your parents, if you go out to dinner with them, particularly your mothers, arms are not long enough anymore, Sasha, to read the dinner menu, right? It means they need glasses. So just keep that in mind. And I start asking someone at the other table, can you hold this? I'm trying to read the salads. It's time to have the glasses talk, right? So there you go. Light waves come in, they get focused through that lens. You need to focus those back on the back of your eye on that retina perfectly. Some people are better at that than others, right? Some of your eyes are too big and some of your lenses don't match, right? So if any of you are nearsighted or farsighted, I don't know if anybody falls into that category, the problem is your lens is not powerful enough or it's too powerful, and it, instead of focusing the image here, it'll focus it here or back here. Unfortunately, that means when it passes through your retina, it's kind of fuzzy. You can think about it like the projector. If you start twisting that knob up there around the lens, the image gets fuzzy, right? Because it's focusing it somewhere other than where the screen is, the screen sort of being the retina. You can correct that, Mary, by just putting another lens in front.
How does lasers actually fix this?
Yeah, so that'll actually correct the shape here of your lens a little bit, and so it'll change. Yeah, so shooting lasers in your eyes sounds like fun. I've got a laser pointer up here in case anybody wants to, save a little money. I don't know what kind of instruments you guys have. I'm willing to give it a try. Paris, are you volunteering or volunteering to hold somebody down while we do it? I'm going to need a couple people to keep them pinned. Either or? Okay. But that's what LASIK will do. It'll kind of change the shape of things out here so that the light waves are focused differently. So not everybody is a candidate for that, depending on the sort of problem the average person. You could talk to a professional about that, or just stop by my office. I have a couple other lasers in there. And I think I have a scalpel even. I've got one of those salon pos patches. We'll cut a hole in it, put it around your eye. You won't feel anything. Nobody's gonna say. Megan, you always look at me like you're in like 2 of my classes, and you look at me like I'm serious most of the time. You think like, I think you might actually try that if I show up at his office.
Well, I am in 2 of your classes, so.
You are. You should get used to this by now. That's the point, right? You should just not be shocked. Phobia, we already talked about that. Very acute vision, color-sensitive area. There is a spot called the optic disc. This is kind of cool. So all of those photoreceptors, they have to have axons coming out of them, right? Because they're cells of your nervous system. In fact, they're the only cells of your nervous system that are visible without busting you open, right? So every time you go to the eye doctor, they shine that light in your eye, They're looking at your nervous system there. It's one reason we know so much about the visual system. It's been accessible to us for a very long period of time. But this optic disc is where all of those axons, all of those fibers punch back through and go back to your brain, right? It actually creates a spot called the blind spot. Now this is not the blind spot of your car. That's a different thing. You can see the blind spot of your car simply by like moving your head. Some people don't do that while they drive. I've seen some of you out there. Right, Scotty, you see these people, they don't turn their heads when they drive. It's a little frightening. It's like, whatever's in front of me is all that matters. But the blind spot is a spot on your retina where you don't have any photoreceptors. And it's sort of like right here, right? Kind of in this little area. If you kind of like think about where it's located. And you can't see anything there. Now, you're not typically aware of this. And the reason you're not, I sort of lied and said we weren't going to think about those eye muscles anymore, but we need to now for just a moment. Your eyes are constantly moving, right? Okay. And even if you think you're looking at something straight ahead, they still twitch a little bit, right? They make these little micro movements, just super small, right? And basically what they're doing when they do this is they're shaking that blind spot around, right? And it's sort of like it's like a Photoshop trick, right? So you're just kind of moving around, filling in that space. So you never really notice that you're missing something from the visual world because it's constantly moving. And your brain just fills in with all the other information around it. All right, other cells of the retina. We do have a thing called a bipolar cell. Don't worry about that too much. It takes information from the photoreceptors to the ganglion cells. Ganglion cells are sort of important. We'll talk about those in a moment. In between your bipolar cells and in between ganglion cells, you have guys called horizontal and amacrine cells. They're basically just telling neighboring cells what's going on, right? They're just kind of letting other photoreceptors know, hey, I think there's some light here. Maybe we should do something about it. Your ganglion cells, though, these guys are kind of important. These are the first cells to fire an action potential. Up until this point, everything we've done has been those receptor potentials, just those graded potentials. At this point, though, we're going to do a real action potential, and we're going to send that out through the optic nerve back into your brain. All right, here's a nice little slice of your eye. Now, everything's in here backwards, right? So your photoreceptors are actually facing the back of your eye. This guy here is called the retinal pigment epithelium. As light comes in, and yes, it has to go through all of those cells, hit those outer segments of your rods and cones, get those guys activated. On some level, you probably think, well, that seems ridiculous. Isn't this going to cause a problem? Possibly. We're not worried about it. What's more important is keeping it close to this retinal pigment epithelium, because that guy's going to help replenish and keep these rods and cones nice and healthy, right? So it's kind of important. We're going to send the signal back out. When we get to these ganglion cells, that's when we fire that action potential, and then we're off to the brain. Here's a test for your blind spot. It's not going to work. If any of you have the book, or you can put this on probably like a phone or a tablet would work well, maybe your laptop. What you do is you close one eye, you look at this dot or this cross, and then you move the blind spot test back and forth kind of slowly. When you get it probably this far from your face, you're going to notice that green spot will sort of flicker and then disappear. It's kind of exciting. Here's another picture of retinal circuitry. Don't worry too much about that. We really just covered it with the last slide. All right. And we already talked about transduction. That was awesome, right? We said in this case we're taking energy, we're taking light, and we're turning it into membrane potentials. Right? I'm not going to ask you about these other things. This is sort of an interesting concept, though. We should think about this. This is called the dark current. If your photoreceptor is not activated, right, if it's in the dark, it's actually going to be depolarized. Now, I know for the most part I've told you things are depolarized when they are activated, right? But this is the reverse, okay? This is the exact opposite. It's going to be hyperpolarized if it's activated. Now your bipolar cell knows what's going on, it's fine. When it gets the hyperpolarized signal from the photoreceptor, it actually depolarizes and causes action potentials in the ganglion cell. If this was going the other way, so if this was in the dark, we don't get any. Now, why would we want to do this? Well, when your photoreceptor is activated, if it was activated and it released a neurotransmitter, there's the potential that it would eventually become fatigued, right? That you would run out of neurotransmitter. And what would happen then? Well, you would be indicating that there was no visual stimulus present, which sounds fine unless you're walking across 20th Street and all of a sudden a significant number of your photoreceptors go, we're all out of neurotransmitter. Guess what? You don't see that car coming around the corner. And Scotty, that was one of these people who doesn't turn their head when they drive and they hit you. Okay? If you flip this and you think, well, what if we shut that down when we get activated? Well, that's easy. And you think, well, what about in the dark? What if I run out of neurotransmitter in the dark? Well, if you're in the dark and you run out of neurotransmitter, you're not looking at anything anyway. You're probably asleep, right, Bethany? I mean, so who cares if your neurotransmitters in your eye go, or your photoreceptors go, well, there's nothing there. You're not seeing anything anyway. So it makes a lot more sense to have that process input. We should think briefly about receptive fields. Each cell in your visual system is only responsible for a small segment of the visual world, right? So if you look out in the world, we can assume that you can kind of see like a 180-degree dome right in front of you, right? And somewhere at every sort of little point out there, there's a cell responsible for that small bit of space. So if we were to plot this like a square, and then we started sort of mapping out receptive fields, you would have all of these little circles. Some would be larger, and then there might be smaller ones inside of that. Sort of depends on what you're trying to do. Not too much to worry about. I guess one more time, we should actually talk about those eye muscles again that move your eyes. There's sort of three types of eye movements, broad types of eye movements that you should think about. One is what we call vergence. So if you were to take your finger, kind of focus on that and move it in and out, your eyes are going to sort of, converge or diverge, right? So they're going to come together and move your eyes that way. We also make what's called saccadic eye movements or saccades. This is when you sort of zip your eyes from one location to the other. All right. You're looking over here and you hear some noise, or you need to follow somebody, you need to look somewhere else. You move your eye over. The third is what we call smooth pursuit or pursuit movement. This is when you are tracking the movement of an object, right? And you can move your eyes smoothly as that object is moving through your visual space. What's really interesting about this is you cannot fake this, right? You can try, and believe me, I have. It's given me more migraines than I care to admit. Because I think every few weeks I'm like, Gentry, I'm going to do it this time. It never works. So if you try to sit there and you imagine, I don't know, like a bubble floating through space, and you're going to follow that with your eyes, think, okay, watch this. And every time you try to do it, it ends up just being a saccade. It's a really super fast eye jump. You can't. Give it a try. I promise you, you'll never be able to do it. Go ahead. We'll take 10 minutes to let you guys do that. Nobody's going to try it. You're going to try it when you get home. You're going to realize it's brilliant, but you can't do it. So, why do we see things better in the fovea? Well, remember that time, a long time ago, I said, hey, on average, each of your cells gets about 10,000 inputs from other cells. And I said some of those maybe get as many as 100,000 inputs, and some of them only get one input, right? So in this case, this guy that only gets one input, well, he knows exactly what happened in that small bit of space, right? So this ganglion cell fired an action potential. That was awesome. It fired an action potential because a photon of light hit this guy. And we know exactly where that is. We know exactly what happened. It's a one-to-one relationship. This ganglion cell here, it's going to fire an action potential. Awesome. Did it fire that action potential because this guy got a photon, Or was it this cell, or this cell, or this cell? We don't know, right? Because all of these cells funnel down to that one ganglion cell. We've lost some of that information, right? And it's not as precise. I kind of think about this like video games, right? Anybody play video games? Go ahead and admit to it. I'll know when we take the first exam who spent too much time playing video games. So, if you played older video games, you probably noticed, that there were pixels, right? So, if you were, I don't know, I'm going to try to draw somebody out of squares. Oh, look at that. That's exciting, right? So everything was sort of pixelated, squared off a little bit, right? As technology improved and they started making the pixels smaller and smaller and smaller and smaller, then you start getting more lifelike and more rounded edges and smooth shapes and whatnot, right? Okay. Same thing in your eye. All right. Optic nerves. Once we leave the eye, right? We have to cross over. We do what's called a partial decussation. Don't worry about this too much. It's going to make your head hurt. Most of the information on one side of your eye, visual information from one eye goes over to the other, but not all of it. Some of it stays on the same side. What's really important is, was it left or right visual hemisphere, or field, right? Not left or right eye. Because if you close one eye, you can still see some things on the other side, right? Go ahead and give it a try, Beth. You're thinking about it. It works, right? Then we send that information back to a guy in the thalamus, called the lateral geniculate nucleus. All right, and that crossover is called the optic chiasm. We should take a moment and talk about this guy called blind sight. Once we leave the lateral geniculate nucleus, we go to a place called primary visual cortex, also known as striate cortex. It's known as striate cortex because it's striped. It has this nice prominent striped pattern, or V1 for short, right? If you want to say it all the time, just call it V1. What's cool about this brain region is this is sort of the first place in cortex where we receive visual information, right? Information has to go here before we go to higher order areas and start making assessments of what are we seeing, where is it located, right? This is very raw, rudimentary information coming in, right? So we're going to get that information. Okay? If you have damage to your primary visual cortex, let's say that guy's knocked out, and I show you an object, and I say, hey, what do you see? You're going to go, I don't see anything, right? And we can repeat this process over and over and over again. I can hold up objects and I don't see it, right? So this individual's blind, right? That makes perfect sense. Primary visual cortex is gone, you're blind, okay? So, what do we do next? Well, the next thing we do with one of these individuals is we set them out in the hallway. We say, hey man, take off walking. And then we start putting things in front of them like trash cans and chairs, right? It's exactly what you should always do to blind people. Put things in front of them. No, don't do that. That's not nice. What's interesting about this particular type of blindness is these individuals can actually successfully dodge objects that are in their way, right? And that's because there are other pathways into other parts of visual cortex besides this primary visual cortex. And there's some visual motion information that goes in through another pathway. We call that blind sight. It's actually pretty cool, right? And there are actually YouTube videos of these folks. You can see these guys like walking down the hall. They're like moving around things. And you'll even ask them like, hey, why did you do that? Like, well, I have no idea. I just felt like I needed to step over to the left a little bit just now because they're not consciously aware of anything, right? But sort of subconsciously they're dodging and moving. It's pretty cool. Later we'll talk about sort of an emotional component of this. These folks are also still sensitive to facial expressions, right? So you can show them smiling faces or angry faces, and they can very, very accurately tell you whether or not the face is smiling or frowning. They'll tell you they don't see anything, and you just kind of say, hey, well, you have to take a guess. And they'll go, I guess it's a smile. And far more often than chance they'll be correct. On top of that, one of the other things they do, if you show them a picture of someone smiling, they actually start to smile a little bit, right? Which is a pretty standard response. If you smile at someone and they smile back, it's a pretty standard response. And so these folks kind of still have that same thing. So it's kind of cool. Other visual information still getting through, even though primary visual cortex is gone. And here's the location of primary visual cortex, kind of at the back of your brain. Here's that optic chiasm we talked about, that crossover point. And there you'll see lateral geniculate nucleus. Not a big deal. All right, we talked about most of this. Once we leave primary visual cortex, we go to this other very creatively named area called V2, right? We sort of continue that process. There's V3, V4, right, V5, and so forth. We'll talk about those brain regions as we need to in just a few moments, because they all do some cool things. Don't worry about that side, it's basically the same thing you just saw. I could spend a very long time telling you about all of these fun layers in the lateral geniculate nucleus. 4 of you are going to be interested in that, 2 of which are going to understand it, 1 of you might remember it. So I think it's probably a waste of our time to do that. It's not going to make a big difference. Thinking about behaviors. The geniculate, why do they call it geniculate? It's knee, right? You can see how it's bent like a knee. Some folks say that looks like a Napoleon hat. I don't know. But I guess now is the time when we should take a moment and watch a movie. Anybody like movies? Yeah, this is a cool movie. This is a movie from 2 guys who won a Nobel Prize. How many of you have ever won a Nobel Prize? I'm not going to ask how many of you have been in the movie. Nobel Prize winners, David Hubel, Torsten Wiesel, right? This is a pretty groundbreaking experiment that I'm going to show you. Sort of think of it as basic information now, right? So what these guys did, they did some work back in the 60s and 70s. They got the Nobel Prize in the 80s for some of this work. What they did is, and you're not going to see everything, so what you're going to see is going to be, so don't worry too much. They actually did recordings from a cat visual cortex, right? So they were taking a metal electrode, they were putting that in cells in the cat visual cortex, and they started showing this cat different things, right? So they would show it visual stimuli, dots, moving bars, all kinds of things. They could map out these receptive fields. You could hear, and in this video you will actually hear the action potentials from those cells. So they've taken that electrical change in potential, they've piped that through an audio monitor, and what's coming out, these kind of cracks and pops, you guys will be able to hear that. Hopefully the speakers are good enough in this room to be able to hear that. Okay? All right. Just take a moment. Visit the magic internet, see if we can find that. We want this one. Alex, you guys go to the back. So what you're seeing here is basically a screen, very similar to this kind of screen. It's got paper on it so these guys can mark. They're taking that fancy thing that the eye doctor uses, and they shine in your eye, right, that ophthalmoscope, and they're making these different sized bars of light. What they're doing is they're flashing that light on and off. Maybe those little pops and crackles just random accidental action potentials that just happen from time to time. But when they get to an area that is excited, you can hear that just a lot. So what you're hearing there are the cells that are responsible for these sort of sections, small section of the visual world, getting excited by the light, by that visual stimulus. It's pretty exciting. What they've marked here is this receptive field has a unique structure. It has sort of these inhibitory flanks on the outside, and then has an excitatory part in the middle. It's oriented at a particular angle, right? This is important so that you can start to determine shapes of objects, right? So how many of you have ever walked through a door? Okay, some of you have done that. That's great for you to understand like the orientation of the sides of the door, right? Because if you weren't able to determine accurately the orientation of the doorposts, then you might have some trouble getting it around. So these cells, they're all tuned for different orientations. Plug that into other things later, and eventually put different orientation bars together to make objects. We're going to do the same thing again. Hear that? This is a different cell. This cell has a different receptive field structure. As you can imagine, there's something that are pretty complicated, so we're going to want to wire these things up in a variety of different ways so that we can accurately determine what's in there. First question you might ask is, aren't there cells up here, right, that are responsible for that part of the world? There are. They just don't have their electrode in that particular cell. They have the electrode in another cell that's going to be responsible for this small segment. This particular cell does not have any inhibitory flanks, so as you make that stimulus bigger, you're not getting any inhibition in stimulus. Let's see if I hit this cell. This one's a cool one too. This one's actually direction selective, so when they move this bar of light in one direction, the cell will get excited, but when they move the bar of light in the other direction, the cell doesn't get excited. So that was just to show you sort of some of the, like how these cells actually are responsible for the world out there. What that sounds like, I think it's cool for you guys to hear the actual action potentials, right, as they're happening. And again, these guys were Nobel Prize winners, not just for that experiment. They did a bunch of other stuff. Basically, they figured out how they respond to these contours, right, or to the edges. Previously, folks just used like little dots. But Hewlett and Wiesel were like, hey, let's make a bar of light and see what happens. The world's not made up of a set of dots, right? You group things into shapes. It's kind of the first step in that process. Don't worry about that. All right. So, we've talked about information as it goes from the eye to the lateral geniculate nucleus and then to primary visual cortex. Still very rough rudimentary information. We've taken this very complex visual scene, kind of broken it down into all these pieces. We've just sent those channels into your brain. What we do later, once we get outside of that striate cortex, is we actually start to put these pieces back together, right? So that we can make a coherent sort of concept of the world that's around us visually, okay? So we have sort of two pathways, right? We have what's called the dorsal stream and the ventral stream. What's interesting about these is they're not just divided sort of spatially in your brain, there's a functional difference as well, right? Things that go in the dorsal stream, so again, think about that dorsal fin, okay? This is going to tell us kind of where, right? So we're interested in location, objects, visual movement, things like that. We're not identifying anything, we're just telling you where it's located, right? Because there is something over there. We don't know what it is. The ventral stream, which goes this way, that's our what pathway? That's going to be for identification of objects. Okay? Now, folks can have brain damage in only maybe one of those streams, right? Someone may have damage to their what pathway, and they can no longer identify certain objects, but they can tell you exactly where they're located, right? Their location ability has not been altered. Some folks can have damage in the dorsal pathway and that where pathway. They have difficulty telling you where things are located in their visual space. They have difficulty moving around in their environment, but they know what's there. They just don't really know where it's located. Right? Things seem out of place or out of order. Don't worry too much about this slide either. More contrast, I'm not going to ask you about that. We should think briefly about color vision, right? Basically, you have three photoreceptors, each sensitive to a different wavelength of light. Okay. And then you have that fourth guy, those are your rods. Again, keep in mind, why do we put blue, green, or red in quotes? It's because that light is not actually blue, green, or red. We perceive it as that color, once we get things into higher order brain regions and we do that processing. This shows you the sort of peak sensitivity for each of those different cone types and the rod. Anybody familiar with color blindness? The most common type of color blindness is red-green, right? And if you look at this, it's actually pretty easy to see why. If there's any sort of alteration in the structure of your red or your green cones, right? We would call these also medium or long wavelength sensitive cones. You can see how close these peaks are, right? And if there's anything that shifts these peaks one direction or the other, you can see that the overlap becomes so much you can't tell the difference, right? So that's why red-green is the most common type of color blindness. It's also most common in males, more common in males than females. The reason for that is related to X and Y chromosomes, right? Females get 2 X's, so if you've got one that's screwed up, that's no problem, you'll just use the other one. If males get an X chromosome that's screwed up, well, too bad. That's the one you gotta use. It's the only one you got. And the genes that control these cones are actually located on the X chromosome, right? So as a female to have color blindness, you would have to have 2 X chromosomes that had a problem at that particular location in order for you to be colorblind. So that's why it's far less common for females. All right, so this is a good color vision test. You should be able to see these numbers. If you can't, I would recommend getting your eyes checked, just to, just for your own safety. Don't worry about any of this. Think about it. Oh, we should think about after images. This is kind of fun. Anybody familiar with after images? So what you can do is probably, let's stare at this, the white plus sign there in the middle of that apple, probably for about an hour. So just stare at that for like an hour and then move your, really you can stare there for 30 or 40 seconds. So if you want to do that, you can stare at that apple for a while. And then you shift your gaze over to that blank space. What you should see, right, is an after image that is instead of a green apple and a magenta leaf, you should see a red apple and a green leaf, right? Did anybody have something similar happen? Okay, great. If it didn't, again, you need to go see a neurologist. Let me sit stare long enough. The reason this happens is if you think about while you're staring at this apple, you're staring at this large patch, right? While you're staring at that, what's being activated? All of those sort of medium wavelength sensitive cones, all of those green sensitive cones, right? What happens after you stimulate these green cones for an extended period of time? They become fatigued, right? And so what happens when you shift your gaze over here to the white, the green is no longer active because they're tired, and all that's going to be left over is red. And if those red cones are just, kind of randomly active where they normally are, you're going to get some red input, right? So instead of like red actually increasing, the red just looks more powerful because the green looks worse, right? How many of you have ever played basketball against your younger cousins? They were like 10 years younger than you, right, Tyler? And you looked awesome because they were 8 and you were 15. And you went home and told your mom about how great you were and you were going to be the next Kobe Bryant. It's a true story, isn't it? I saw it on your Facebook page. Don't worry about it. I don't even know if you have a Facebook page. Don't tell me. I really don't care. It's going to take up space for something important. And one of these days, somebody's going to say something I'm going to forget how to get home. I don't want that to happen. It's a finite amount of space. I got to put important things up there. Questions about this? It's kind of cool. This also explains the, you guys familiar with the waterfall illusion? Or, so if you stare at something for a long period of time moving in one direction, and then you look at something that's not moving, it'll look like it's moving in the opposite direction. It's also because they're motion sensitive cells, and if they're all getting activated, if all the, let's say the rightward motion cells are getting activated, and you look at something static, all that's left are those leftward motion cells that seem to be a little bit active, even though they're not. They're really getting activated, but because those rightward cells are no longer really doing anything, that difference in activation is what's important. And don't worry about this. All right, so let's talk about some studies with humans, right? Everybody loves humans. So, again, because we're not allowed to like scoop out chunks of people's brains and kind of see what happens, we have to wait on someone to have some sort of damage, right, to their brain that can occur through a stroke, head injury, neurodegenerative disorder, or we can do fMRI studies, right? You can do those as well. They're fairly expensive, but you can pull those off. So here's a study, a couple studies where folks were looking at an area called V8, and they actually figured out this is sort of the color-sensitive brain region, right? So this is one of the regions, one of those higher order extrastriate cortical regions that handles color vision. So they gave folks an fMRI. You sit there, you look at these colored images, and then you look at images that are monochromatic, and then you subtract off, which areas are activated both times, and whatever you're left with, those are the brain areas that are active or responsible for processing color information. So they labeled that as V8. This particular study, pretty impressive, because it actually had 92 people, which is a pretty high number for some kind of clinical study like this. They were not able to see color at all, but there was not a problem with their retina. That's the first thing you have to check, right? Because if somebody says, oh, I don't see any colors, you want to check their retina to see if they have cones. All of these folks had functioning cones, right? And so everything was good up until you get to V8. All 92 of these individuals had damage to the brain region V8, and so that's why they were not able to process color information. Kind of exciting, right? How many of you would like walking around and everything was like shades of gray? It'd be awful, right? You don't think about it, you're like, oh, well, you know, who cares what clothes I'm wearing? I can't tell if it matches or not. That might be embarrassing. What would be really bad is food, right? Because everything's going to just, I don't know. I read an account of a guy who had this problem, and he said everything looked like it was the same color as a rat. And so, like, he's just eating, it's awful. And he said the only foods he could really eat were foods that were white or black, because those were still things that came through to him. So he would eat like black olives or yogurt, and those were like the only things he would eat. Because everything else, the color didn't look right, and it just made him sick. You don't think about how important color vision is, right? There you go. I want to eat a hamburger the same color as a rat. Just sounds gross, right? You need that bright red tomato and the green lettuce. Big slice of yellow cheese. I know. All right, and so there you go. There's a little bit of kind of showing the damage down there of this particular individual that this person actually lost form but not color. So they could still see colors, but they couldn't see what was going on in the world. So they would just see like blobs of color out there, but they couldn't tell you what they were looking at. And this was area V2, which is kind of interesting, just a little bit away from area V1. Really important for that spatial information. I'm not going to ask you too much about spatial frequency. Although this is kind of cool, right? How many of you love Abraham Lincoln? That's like a universal question, right? It's like nobody's going to answer no to that. So these are both images of Abraham Lincoln, in case you didn't know that. But they have different amounts of high or low frequency information, and you can actually, if you were to back away far enough, you might get a clearer image. I don't know, Paris, are either of those fairly clear to you or not? No, you're not far enough away. Eventually you can get far enough away and that information will sort of blend out. You guys ever seen the one? I saw one that was Albert Einstein and Marilyn Monroe. It's kind of an interesting, it was really interesting juxtaposition of two individuals there. But when you were fairly far away, it looked like Marilyn Monroe, and so you're like, oh, I'm going to walk up to that and see what it is. And as you got closer, it turned into Albert Einstein. It was pretty cool, actually. You should get one of those, put it on the wall at your house. I think about that. What if you could do like two other images? What if you had an image that was really like inviting on your front door like that, right? You know, that's what everyone's going to have to be, one of those individuals. And then all of a sudden, when people get up close to it, it turns into something that will just really make people run from your house. I'm going to work on that. I think there's a market. So these are other sort of areas that handle form. I'm not going to ask you about those. But you can do similar studies. This is actually a monkey brain. Okay. So now we should talk about some interesting areas still in that extrastriate cortex or beyond primary visual cortex. There are folks who have damage to a variety of these brain regions, and they will sort of lose the ability to identify objects, right? Sometimes they lose the ability to identify faces. Okay, that's kind of a big one, right? You definitely want to be able to identify people's faces. Right? You don't want to get mixed up there. You tell the wrong person the wrong thing, it's just, trust me, it can get dangerous. But there's an area called the fusiform face area. And in humans, this area really processes faces, right? So what's kind of interesting about faces is they all sort of look the same, but they all sort of look different too, right? So if you think about faces, they all sort of have the same components, right? Noses, ears, things like that, eyes, whatever. And so if you were thinking about this, averaging this out, right? If you were like a, I don't know, let's say you were a cricket or a frog, or who likes barn owls? I don't know. Doesn't really matter. If you're some other sort of species, right, you're looking at people's faces and you're like, oh man, all those people look the same. They've all got that like kind of thing in the middle of their face, kind of vertical, and then they've got like some kind of horizontal thing. There's like two balls that keep moving around, right? It's all sort of the same. Now to us, you think, well, like nobody's face is the same, right? Even if you know identical twins, you're like, I can definitely tell those two people apart because of these like minor, changes in their faces, right? And so to us, because we're experts on this facial identification, we think all faces are really different, but in fact they're all pretty much the same when you average it out, right? So what's interesting about the fusiform face area is not only does it work for faces, but it also gets excited for sort of other like expert evaluations, right? And so if any of you are sort of, you have like a hobby, you're really interested in something, something where if like someone who's not interested in it would look at those objects and they would go, well, they all look the same to me. But someone who is an expert would notice all like those, those minor differences. Then, your fusiform face area is going to get excited for that. Anybody into musical instruments? Yeah, right. So maybe you collect those, 6-stringed instruments, that are like this long and have like a bulge on one end and like a long part you hang on to with the other, right? Also known as a guitar. To a lot of folks, you look at those like, man, they all look the same. There's a blue one and a red one. But to an expert, you might think about, something on the fretboard. You might think about the different pickup orientations. You might think about, this is this brand or this is that brand or this is actually made from this year to this year of this brand. You can tell because of, the thickness of the neck and the shape of whatever, right? But to somebody who's not an expert, they're going to go, well, if you move your fingers across it, does it make noise? Then it's a guitar. That's kind of the end of the story. But for those other folks, the fusiform face area would get excited about that if you're an expert on that. Does that make sense? Yeah, it's kind of cool, right? You also have a thing called the extrastriate body area. This is cool. It gets excited by body parts, right? I don't know if you ever like randomly seen body parts somewhere. But it's going to own up to that. But it gets excited by body parts, right? So there's a brain region that's specifically tuned for human body parts. Pretty important. How many of you have ever seen human body parts? Yeah, like everybody has, right? So you spend a lot of time looking at these things. And so here's your extrastriate body area. If you show them body parts or headless bodies, images of that, they get pretty excited. Show them faces or assorted objects, not so much. The fusiform face area, on the other hand, will get pretty excited when you show it faces. Somewhat excited if you show it headless bodies. I think it's anticipating a face, right? Like, there's nothing there. That's something weird. Let's get excited about that. Don't worry about the very hippocampal face area right now. The other cool thing about the fusiform face area is it actually changes over the course of development, right? How many of you were once like 7 years old? I'm assuming most of you were 7 years old at one time in your life. At that time in your life, how many faces had you seen compared to how many faces you've seen now? The correct answer is not as many, right? Doesn't really matter, like an actual number, but not as many. So if you're a child, you've seen fewer faces than adults, right? As adults, we've seen a considerable number more faces than children. So here's like an 8-year-old kid. Now, I know somebody's going to say, well, like, hey, aren't kids' brains different size than adult brains? Absolutely. They did some fancy math here to equalize the size of the brains, right? So we're not kind of getting anything distorted here. Here's the fusiform face area in that 8-year-old child. Here's the fusiform face area in an adult. Look how much larger that that brain region is. So as you become an adult, you kind of gain some expert there in facial identification, right? As you get older, because you've seen more faces, you can see those minor differences better. You do a better job. We can think a little bit about depth perception if you want. I think it's that important. You have two eyes. They're not stacked on top of each other, right? And that's really great if you want to know where something is located and how far away it is from you, right? You use all kinds of cues to figure that out, right? So think about perspective, relative retinal size, something's bigger or smaller on your retina, details, things like that. I've always had these questions, like I wonder if somebody with like wider set eyes has better spatial vision, right, or depth perception, right? You ever think about that? And then I thought, well, probably not, because you know you've got those other components in your brain; they're probably not adjusted for that. So, something to think about, though, right? If any of you have really close set eyes, you should find someone else with close set eyes and try to have a bunch of kids with close set eyes. And if any of you have wide set eyes, we'll do the same thing, right? You guys have a bunch of kids, and then we'll test them later for their depth perception. Seems reasonable. I don't know why people don't think about these things, right? I mean, this is brilliant, right? I mean, if you wanted your child to be great at judging distances, right? Maybe you want to start measuring distance between eyes of your partners, right? Nobody thinks about that. These small things can make a difference in your life. All right, we already talked about human visual. Here's basically orientation, selectivity. Oh, optic flow is kind of cool. How many of you have ever moved through space? Yeah, you guys did a great job on that. So if you, this is like complex motion, you're walking down the hall, right? So things are kind of at this point and they kind of move out around you. Super awesome. There's something called ekinetosia. This is actually pretty cool. So if you have damage to a brain region called B5, you actually cannot perceive movement. So all you're seeing are like static images all the time, right? Can't perceive movement. Something moves, it disappears, right? So if you're standing still, they can see you, and then you start walking, and then you're gone. You still hear your voice. It's like this disembodied voice floating across space, and then you walk over here and you stop, and all of a sudden, wow, you appeared again. That's pretty interesting, right? I think the moral of the story is don't hit yourself in the head, right? Because you could cause brain damage, and you could lose the ability to see motion. I think that's reasonable advice, right? Let's see. Visual motion. Here's all that list of brain regions. Tells you what they do, where they're located, and so forth.